Hello! In this video, our group will present the methods for determining spinal reflexes and reactions in humans, along with the experiments involved. The objective of this experiment is to measure and compare reaction times to visual and tactile stimuli. To determine whether reaction time improves with practice, and to analyze the role of the spinal cord and brain in reflex actions. A reflex is an involuntary and predictable response to a stimulus. But what exactly is a stimulus? A stimulus is anything that triggers an action, whether it's a sudden movement, a sensation, or even an emotional response. Reflexes work through specialized neural pathways called reflex arcs. These arcs connect through the central nervous system, the brain, and spinal cord to produce quick, automatic responses. While most reflexes involve the brain, some rely solely on the spinal cord. This process operates as a negative feedback circuit and consists of three main components. Sensory component, afferent neuron, detects stimuli, converts it into an electrical signal, and sends it to the central nervous system. Integrating center, interneuron, located in the spinal cord, this acts as the processing hub, analyzing the signal and determining the appropriate response. Efferent portion, motor neuron, carries the response signal from the inner neuron to the effector stipically muscles or glands. For example, in the patellar reflex, it activates muscle movement, while in the salivary reflex, it stimulates gland secretion. In short, reflex arcs allow our bodies to react swiftly, protecting us from harm and maintaining balance without us even thinking about it. Now, let's explore the ways to determine spinal reflexes and reactions in humans, as well as the experiments involved. In this experiment, we are investigating the patellar reflex knee jerk to understand how reflex actions occur and the role of the nervous system in involuntary responses. The aim of this experiment is to observe the involuntary response of the lower leg when the patellar tendon is tapped, and to analyze the function of reflex arcs in human physiology. The procedure involved seating the subject on a bench or table with the lower leg hanging freely to ensure the quadriceps muscle was relaxed. The patella, kneecap, was located, and the reflex hammer was gently tapped on, or just below the patella. Any extension of the lower leg was observed and noted. This experiment helps demonstrate the monosynaptic reflex arc, where sensory and motor neurons work together to produce an immediate response without involving the brain. The results provide insight into neuromuscular function and reflex testing in medical assessments. In this experiment, we are measuring the Achilles tendon reflex. The subject's shoe was removed, and they were instructed to kneel on a chair with their feet hanging over the edge. With the foot relaxed, a small tendon hammer was used to lightly tap the Achilles tendon. The stimulation was repeated at approximately 2 taps per second. In this experiment, we are determining the subject's reaction time to sight. Both the experimenter and the subject were given a stopwatch and instructed to start them at the same time. The experimenter stopped their stopwatch at a random moment, and as soon as the subject noticed, they stopped theirs as well. The time difference between the two actions was recorded to measure the subject's reaction time. This process was repeated 10 times, and the average reaction time was calculated. In this experiment, we are measuring the subject's reaction time to touch. Both the experimenter and the subject were given a stopwatch and instructed to start them simultaneously. The subject's eyes were closed to eliminate visual cues. At a random moment, the experimenter stopped their stopwatch while simultaneously stepping on the subject's foot. As soon as the subject felt the stimulus, they stopped their stopwatch. The reaction time was measured and recorded. This procedure was repeated 10 times, and the average reaction time was calculated. The experiment demonstrated that reaction time to touch is slightly faster than reaction time to sight due to differences in neural processing. Tactile stimuli are processed more directly through spinal pathways, resulting in quicker responses, whereas visual stimuli require additional brain processing, leading to a slight delay. The results also showed that reaction time improves with practice, as the subject responded more quickly in later trials. This improvement suggests that repeated exposure enhances neural coordination and response efficiency. Additionally, the distinction between spinal and cerebral reflexes was evident, with spinal reflexes allowing for faster responses by bypassing higher brain centers. Overall, the findings highlight the adaptability and efficiency of the nervous system in processing different types of stimuli and refining responses through experience.